So after the successful completion of the fifth chapter, that is in second PUC, let me start with the sixth chapter, the biggest chapter in the second PUC and very important chapter for your NEET examination, that is the molecular inheritance, that is molecular basis of inheritance, molecular basis of inheritance. That is the biggest chapter, that is in second PUC and equally important for your NEET examinations. So it begins with the nucleic acids. Nucleic acids are the hereditary materials discovered by Friedrich Miescher in 1869. So these years also very important. In 1869, Friedrich Miescher discovered the nucleic acids. There are two types of nucleic acids, DNA and RNA. The sugar present in DNA is a deoxyribose sugar, in RNA it is the ribose sugar. So these are the nucleic acids. And coming to the DNA, the nitrogen bases present in DNA are A, G, C, T, adenine, guanine, cytosine, thymine. AG are called purines, adenine, guanine. CT called pyrimidines. They are larger than the purines. The purines are larger than the pyrimidines. So they have heterocyclic nitrogen bases. These are all the nitrogen bases present in the DNA. Adenine, guanine. Structurally, actually, to write the structures and all, structurally adenine is the 6th amino purine, the 6th amino purine. 2-amino-6-oxypurine is guanine. 2-oxy-4-amino pyrimidine is cytosine. 2,4-dioxy-5-methyl pyrimidine is thymine. So these questions will be asked in the CET as well as NEET. Many times, repeatedly they are asking about the structures. So that is uracil. 5-methyl uracil is 5-methyl uracil is nothing but the thymine. 2,4-dioxy 5-methyl pyrimidine. 2,4-dioxy 5-methyl pyrimidine is thymine. Like this they may ask. Once again I repeat. 6-amino purine is adenine, and then 2-amino-6-oxypurine is guanine, and 2,4-dioxy pyrimidine is uracil. 2,4-dioxy 5-methyl pyrimidine is thymine. AGCT are the purines and pyrimidines in DNA. Thymine. In RNA, thymine is replaced by uracil. 2,4-dioxypyrimidine. Once again, 6-amino purine, and then 2-amino-6-oxypurine. Then the reverse modulation. 2-oxy-4-amino pyrimidine is cytosine. 2,4-dioxypyrimidine is uracil. 2,4-dioxypyrimidine. 5-methyl pyrimidine is the thymine. So this is about the structures. A nitrogen base with sugar part, sugar moiety called nucleoside. Nucleosides of DNA are adenosine, guanosine, cytidine, thymidine. A nucleoside with phosphate molecule called nucleotide. Nucleotide. So nucleotides of DNA are adenosine monophosphate, athwa adenylic acid, guanidinic acid, cytidinic acid and thymidinic acid. The phosphate molecule present is H3PO4. H3PO4, and the bond formed is called ester bond. COO bond is established between the nitrogen base and sugar. Hence, they are the polynucleotide chains are the nucleic acids. Nucleic acids are the polynucleotide chains, and they are the hereditary materials, that is DNA as well as RNA. So the model of DNA that remained elusive until the elucidation of X-ray crystallographic study of that means the model of DNA remained elusive until the elucidation of extracrystallographic study of DNA by the Maurice Wilkins and Rosalind Franklin. After this, the James Watson and Francis Crick in 1953 proposed the model of DNA. According to this model of DNA, DNA is a double helix consisting of two strands which are arranged spirally and antiparallelly. Body or the framework composed of phosphates and sugars. The steps in between them composed of nitrogen base pairs. Base pairs of DNA are ATGC. Adenine binds with thymine with two hydrogen bonds or vice versa. Guanine binds with cytosine with three hydrogen bonds or vice versa. This kind of binding is very specific. The space between the internucleotide space between 3.4 angstroms given there, and here it is given 0.34 nanometers. That is the internucleotide space between the nucleotides. And coming to the number of nucleotides in different organisms, say for example a bacteriophage called pi into 174. The number of nucleotide bases here is 5386. And then the number of base pairs, nucleotide base pairs in bacteriophage, lambda bacteriophage. First one, a bacteriophage called pi into 174. There the number of nucleotides 5386. Whereas in case of lambda bacteriophage, it is 48,502, 48502 base pairs are present. Coming to the Escherichia coli, here it is number of base pairs 4.6 into 10 to the power of 6, and the number of base pairs in human genome, that is haploid set of chromosomes, 3.3 into 10 to the power of 9. And the length of DNA, that is very important. The length of DNA in the human cell, that is very important. Sometimes sums will be given, little problems will be given with reference to this. The length of DNA in the human cell is 6.6 into 10 to the power of 9 into 0.34 into 10 to the power of minus the inverse 9. That equals 2.2 meters. That is very, very important. That is very important for the NEET examination. So, then the Erwin Charagaff, according to Erwin Charagaff, amount of purines will be equal to amount of pyrimidines. That is Ag equals Ct. 50% if Ag present, 50% Ct present. Wherein the amount of adenine will be equal to amount of thymine, amount of cytosine will be equal to amount of guanine. So this is also very important. A simple sum, a problem will be given for this. If, for example, adenine is 15%, thymine also 15%, then the guanine and cytosine remaining 35 each, 50 equals 50. This is called Charagaff's base rule proposed by Erwin Charagaff. That is about the DNA, and then the DNA is a double helix consisting of two strands which are arranged spirally anti-parallel. So towards one end, the phosphate group binds with the sugar that is at carbon 5, that is 5' end, and anti-parallel towards the opposite, in the opposite strand, the reverse type sugar, there the phosphate binds with the carbon atom 3, hence it is 3'. This is also very important as far as the model and the structure of DNA is concerned. And see the diagrammatic representation of all these things, that will come to know, that will be very easy for you to remember. Next to this chromosomes. Chromosomes, a chemical composition of chromosomes, DNA and histones. DNA and histones. DNA along with octamer of histones forms the nucleosomes. The nucleosomes, they form the repeated units in chromatin. That is very, very important. Units of chromatin, nucleosomes. Nucleosomes composed of DNA and octamer of the histones made up of lysine and arginine in eukaryotic organisms. In prokaryotic organisms, there the DNA wrapped around some proteins not given, which are whether they are histones or not given, only the proteins given. That is very important. That is very important. There the DNA in prokaryotic organisms wrapped around some proteins. Here the DNA coiled around octamer of histones made up of lysine and arginine. This is very important in eukaryotic cell. And then the number of base pairs in the DNA, about 200 base pairs is what we care. That's important. As it proceeds from the prophase and then metaphase, these chromatin along with the chromatin fibers together constitute chromosomes. Only the chromosomes type we can see that is in the metaphase, that is important. And then in addition to this, for these chromosomes some other proteins, additional proteins are added called NHC, non-histone chromosomal proteins. This is also very important. And then there are two types of non-histone chromosomal proteins: heterochromatin and euchromatin. Heterochromatin, very important. Code word on HD Devegowda. HDD Devegowda, sir. HDD andre heterochromatin, densely coiled and darkly stained, but transcriptionally less active. HDD. Heterochromatin, densely coiled, darkly stained, transcriptionally inactive. Opposite euchromatin, less coiled, less stained and transcriptionally active. This is very, very, very important. And then coming to the next aspect, that is these nucleic acids are discovered in the year 1869. But up to the 1926, with the discoveries of the Sutton, John Greger, Mendel, T.H. Morgan, did not explain which is the molecule that is the genetic material. What molecule is a genetic material, nobody knows until the 1926, though the nucleic acids are discovered in 1869. So many things collected from the Mendel, then Sutton and T.H. Morgan, and the scientists, they wanted to know which is the genetic material. And only in 1928, Griffith did a meticulous experiment called transformation experiment. This is very important. During the transformation, he wanted to know which material is the genetic material, which molecule is the genetic material. So he took the two strains of Streptococcus pneumoniae bacteria, Streptococcus pneumoniae, two strains, S type and R type. S type capsulated polysaccharide coat it has, and it is virulent. R type no polysaccharide coat and it is non-virulent. So when R type injected, rat not affected. When S type injected, because it is virulent, rats killed. So killed many mice or rats. And when heat killed S type injected, rats not affected, not killed, because it is heat killed S type. When a mixture of heat killed S type and R type injected, many rats were killed. So this is, and they wanted to know R-type obtaining something from the S-type, which is heat killed. R-type become virulent. That experiment is called transformation experiment. It is the R-type, R-type obtaining something from the heat killed S-type became virulent. And what is that material? This R-type obtained from S-type does not know. He doesn't know. Then after these experiments, in 1933 to 44, Oswald Avery and Colin MacLeod and Maclean M.C. Carty. So these three scientists, Oswald Avery and Colin MacLeod and Maclean M.C. Carty, they did experiment to prove which material transformed the R-type into the virulent. So they conducted experiments on the S strain bacteria. That means they took three enzymes, one enzyme proteases, RNases, DNases. When proteases injected and transformation takes place, because proteins are not the genetic material which is responsible for transformation. When RNases, which destroy RNases injected, transformation continued. When only DNases injected, transformation will not take place. Thus they proved that the DNA is the hereditary material that has been transferred from the heat killed S-type to R-type. This is the continuation of the experiments of Griffith experiment, that is transformation experiment. Next to this, unequivocal proof for the genetic material comes from the experiments of Hershey and Chase. Hershey and Chase, Alfred Hershey and Martha Chase. Alfred Hershey and Martha Chase. They did experiments in 1952 for which they used two types of bacteriophages. A virus infecting bacteria called bacteriophage. One bacteriophage, it was radioactivated with the P32 and another one radioactivated with the S35. So P32 and S35. When these two bacteriophages infected, that is get attached to the Escherichia coli. And then what happened? So this one bacteriophage with the P32, that is radioactivated, here the nucleic acid, as it contains phosphorus, it gets radioactivated and radioactive nucleic acid by the P32. And here the protein coat, as the protein coat consists of sulfur, the S35. So that protein coat gets radioactive with this. So slowly they injected both the DNAs in different Escherichia coli. And after this, the infection, next the blending stage, and in the final stage, what happened? So when this mixture, from this mixture, the bacterial cell into which the P32 injected, that bacteria shows radioactivity. But here only the nucleic acid injected into doesn't show the radioactivity, and the newly formed progeny will have both protein coat and the genetic material, that is DNA. This shows that DNA is the hereditary material, as the nucleic acid consists of radioactivated with P32 remained inside and produce the new progeny, and such bacteria only radioactivated. Thus unequivocal proof for this one comes from the experiments of the Alfred Hershey and the Chase. So this is about the experiments which proved DNA as the hereditary material. But DNA is the very good genetic material because of many reasons. So it is DNA is very stable because of several things. So the the plane of one base pair stacks over the other. Plane of one base pair stacks over another base pairs, that gives stability to DNA. And then the double-stranded nature also offers stability to DNA. The presence of hydrogen bonds, adenine binds with 2 hydrogen bonds, guanine binds with cytosine 3 hydrogen bonds. This also confers, and the presence of thymine also. These are the 4 things which makes a DNA stable. As it is the stable, it is very good genetic material, because the important characteristic feature of genetic material, the genetic material should be stable, and it is the DNA which is self-synthesized and undergoes replication. Why DNA is preferred as the genetic material? Because it is self-synthesized and undergoes replication. That is very important. And then the second thing is it is stable because of plane of one base pair packed above the other one gives stability, and the presence of hydrogen bonds and double-stranded nature is very important too. And then this is very important 3, and the presence of thymine also provides confers DNA the stable, undergoes less mutation and follows Mendelian laws. That is also very important. And then the other thing, less mutation and less active, less catalytic. Hence it is the very good genetic material for storage of genetic information. Whereas the RNA is labile and degradable because of the presence of OH. This is very important. It is unstable. More catalytic. Undergoes mutation very easily because it is single-stranded. Undergoes changes. Whereas the double-stranded DNA resists changes, and hence it is more catalytic and then very active. Undergoes immediate mutations. This is what we see in the viruses, immediate mutation. COVID viruses, many has undergone many mutations. This is because of the presence of OH, that is in the ribose sugar in the viruses, because the viruses this RNA is labile and degradable. This is all the various reasons, and it is the first genetic material is the RNA, because and then RNA is genetic material because many things like transcription, whether splicing and all these things involved around RNA. RNA is the genetic the first genetic material, but it is very good genetic material for the transfer of genetic information, not for the storage of genetic information. That is very important. And the Francis Crick proposed central dogma of molecular biology. That is, the DNA giving rise to RNA, RNA gives rise to proteins. This is very important. The central dogma of molecular. A reverse of central dogma of molecular biology, RNA giving rise to DNA, can be seen in few viruses. For example, HIV. That is called reverse transcription. And these are some of the differences between DNA and RNA. Next, let me start with the part 2. Let me start with the replication, and then the transcription, translation, genetic code, protein synthesis, characteristic features of genetic code. That is one aspect, and the remaining aspect in the part 3. As this is the biggest chapter, ಅದನ್ನು 3 ಭಾಗಗಳನ್ನಾಗಿ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಚೆನ್ನಾಗಿ ಓದ್ಕೊಳ್ಳಿ, ಯಾವುದು ಸೆಂಟೆನ್ಸ್ ಬಿಡಬೇಡಿ. These are all the very important things. Please subscribe.